Hello everyone, my name is Katie Jamison and I have recently finished my MSc in Paleobiology at the University of Bristol in the UK. Supervised by Dr. Andy Frass and Dr. Ben Moon, I am presenting to you my current research project that I recently submitted my master's thesis on. This project is titled Diversity Dynamics of Microfossils across the Cretaceous Paleogene and Eocene-Oligocene Transitions. So let's kick things off then by discussing why microfossils are important. Microfossils are abundant, well preserved in the fossil record, and are highly diverse, which fluctuates over time. They are inextricably linked to their environment, and so any global perturbations within the environment are reflected within microfossil assemblages, making them excellent for high-resolution diversity reconstruction. So I will briefly introduce you to the 4 different microfossil groups I have investigated. Foraminifera and nanofossils are predominantly calcareous. Planktic foraminifera are cosmopolitan, a lot larger than nanofossils, and their shells are commonly divided into chambers that are added during growth. Nanofossils are mainly autotrophic and restricted to the photic zone. They build their structure by surrounding themselves with a scale-like plating made of calcite called coccoliths. Preservation of calcareous organisms are dependent on the carbonate compensation depth, or CCD, dictating the carbonate saturation of the ocean. The CCD is controlled by various environmental influences, and sediments below this threshold are devoid of carbonate. In contrast, diatoms and radiolarians are siliceous in composition, forming ornate skeletons. Diatoms are mainly autotrophic and form hard but porous cell walls called frustrules, which vary dramatically in size and structure. Radiolarians are largely non-motile, buoyant organisms, occupying a significant vertical range of the ocean, and commonly have a diameter of 100 to 250 micrometers. Biogenic silica formation depends on the balance between organic productivity and other controls such as dissolution, upwelling, and accumulation rates. Undersaturation of oceanic silica can initiate the dissolution of silica organisms in the water column and in marine sediments. So what is it that we'll be testing? So firstly, that microfossil diversity evolution changes through time. Then, due to the nature of an impact extinction at the KPG, I expect a rapid onset extinction with high extinction rates and low speciation rates. On the other hand, the Eocene-Oligocene transition is an example of long-term climate change, and I expect that it will be a period of gradual biotic turnover with both high extinction and speciation rates. To achieve this, I use a novel method called pyrate, which independently teases out extinction and speciation rates from global occurrence data. I then evaluate whether speciation or extinction is driving the diversity change across these extinction events, as well as evaluate selectivity of test composition and size within foraminifera across these transitions. So I thought I should quickly address what pyrate is and why I'm using it. Pyrate presents a novel method of estimating diversity in micropaleontology. Pyrate works within Python, performing macroevolutionary analyses on large fossil occurrence datasets, including all possible data to estimate speciation and extinction rates, producing plots like this one here on the left. It performs analyses within a hierarchical Bayesian framework, producing millions of generations of different differing data combinations to find the best model that explains the data and accurately determines the lifespan of a species. Moving on to the methods then. Firstly, I collected data from the Neptune database, which is a micropaleontology database holding thousands of occurrence records of different microfossil groups, pulled from scientific drilling expeditions. I then tidied up this data within R. I tested for model sensitivity to see how sample age uncertainty impacts overall model uncertainty. There is no discernible difference between the plots of 4 different age errors, and therefore sample age uncertainty does not significantly impact upon the results. I experimented with the 3 main model types within pyrate. I ran these different models on all 4 microfossil groups, as well as groupings of calcareous and siliceous taxa. I then completed maximum likelihood tests to see which model fit the data best for each group. The models with the lowest AIC score, which in most cases was NHPP, except within diatoms, are the models used to present my results. Finally, to test selectivity of foraminiferal size across major transitions, I downloaded the Cenozoic planktic foraminifera phylogeny from A's et al. And used foram size data collected by Andy Frass, and a phylogenetic least squares regression was completed in R. The PGLS method was adapted from Putik et al. 2017 and Allen et al. 2019. NSB data was compared to the planktic foraminifera phylogeny to correct for misidentifications and problematic taxa. Size data was tested against the number of extinctions, and a phylogenetic signal in the pattern of extinction was tested for to inquire whether clades preferentially go extinct together. Trait data was included with the phylogeny to statistically test whether size has a selective advantage across major transitions in a PGLS, looping through time bins from 64 million years to present day. Since this study covers 4 different microfossil groups spanning from 140 million years to the present day, I'm presenting a condensed version of my results, excluding much of the Cretaceous and the Eocene, so I'm basically focusing on the KPG, PETM and EOT. I'm happy to answer any questions regarding my results following this presentation. I should also note that Pyrate is designed to produce extinction and speciation plots like the ones shown here, with a frequency of rate shift graph associated with it. The dotted lines on the graph represent Bayes factor thresholds of positive and strong significance. So let's take a look at the Cretaceous-Paleogene extinction event. The KPG transition is characterised by an asteroid impact and Deccan Traps volcanism associated with extensive marine and terrestrial extinctions. Calcareous records demonstrate a large decline in diversification, with foraminifera experiencing the most significant extinction in their geological record, supporting my proposed hypothesis. This is likely the result of aerosols, soot from wildfires, and ejecta sparking a short impact winter, reducing climate forcing and ceasing photosynthesis. Nanofossils experience a large extinction coupled with a significant change in speciation, likely the result of aberrant, opportunistic disaster taxa, originating in regional blooms shortly after the impact. Corresponding with species longevity plummeting, these taxa would have been short-lived, colonising new morphospace before diversity began to increase again. Diversity increase coincides with the origination of photosymbiotic taxa. Danian taxa were unusually small, shown here with a typically negative excursion in PGLS at 62 to 60 million years. Smaller, more angular forms could provide a selective advantage for harbouring symbionts in the first photosymbiotic taxa. Radiolarians, on the other hand, demonstrate continually high speciation rates for the late Cretaceous and initially across the KPG, while extinction rates remain low. Nutrient-rich waters and cool upwelling zones in the earliest Paleocene are likely to have advanced siliceous plankton speciation, promoting the accumulation of deep-sea biosiliceous deposition. This contrasts the hypothesis proposed for the KPG transition and suggests sea surface temperatures, nutrient input and ocean circulation patterns are integral in radiolarian radiations. The Paleocene-Eocene transition is indicated by an abrupt onset of a negative carbon isotope excursion. Origination of this event is debated, yet common theory suggests volcanism or extensive dissociation of methane hydrates, leading to intense warming and ocean acidification. Calcareous microfossils indicate significant rate changes in extinction and speciation, a likely consequence of dissolution of cold water taxa from increased CO2 and speciation of warm-dwelling taxa. Despite extinctions, planktic foraminifera diversity increases overall, a probable response to deepening of the CCD. Acidification at the PETM is of the same magnitude as at the KPG, yet diversification increases overall, demonstrating that acidification is unlikely a main control in calcareous extinctions, while sea surface temperatures and the CCD fluctuations are likely to impact calcareous diversity more. Siliceous extinction increases significantly, net diversification plummets and species longevity declines simultaneous to the onset of the PETM, owing to an extinction of cold water taxa and rapid proliferation of tropical siliceous taxa. This contrasts the expected turnover, likely not reflected due to the low sampling and preservation rates, particularly in high latitudes during this period. Net diversification finally increases again simultaneous to the early Eocene climatic optimum. At the Eocene-Oligocene transition, continental ice sheet growth and a global oxygen isotope shift was triggered by orbital forcing, influencing a sea surface temperature drop of 3 to 5 degrees Celsius. Hypothesised as a turnover event in all fossil groups, it was expected that EOT diversity would reflect a move to cold water taxa. Many studies attribute the event to a period of extinction in calcareous groups, whereas siliceous groups have a large turnover and show significant increases in speciation rates. Yet the results here present a very different trend. Reorganisation of the oceans and climate interactions is likely to have changed productivity and diversity in high latitudes, which are the undersampled regions within the NSB data in both calcareous and siliceous groups. Consequently, Pyrate is unlikely to have registered these changes in diversity. Furthermore, the CCD during the early Oligocene deepened by roughly a kilometre within the tropical Pacific, simultaneous to ice sheet growth and sea level decline, moving deep water deposition from opal-rich to carbonate-rich sediments. Across the EOT, increased extinctions of larger foraminifera occur and reduced test sizes are recorded at 36 to 34 million years ago. A reduction in body size from ancestor to descendant is present pre-EOT and unlikely to be influenced by phylogenetic relationships. This can be categorised as the Lilliput effect. Causes for this phenomenon are unknown, but the dwarfed forms may signify a selective advantage, such as inhabiting and exploiting a niche space, inaccessible to the ancestor, allowing them to survive chemical or physical oceanic perturbations that triggered extinctions of their larger predecessors. Similar results were observed in the Miocene. Larger taxa are noted at 18 to 16 million years, simultaneous to increased CO2, higher temperatures, intermittent weathering and higher productivity. Then between 14 to 13 million years, similar to cooling at the EOT, temperature decline triggered by orbital forcing promotes a selective advantage for smaller taxa. These results suggest sea surface temperatures are integral to foraminiferal size selectivity. My next endeavour for this project is to infer what macroevolutionary influences were at work during these periods of diversity change. I hope to do this using paleogeny, a paleoclimatic model to associate diversity change with changes in environmental parameters such as temperature and salinity. I can then combine paleoclimatic data with advanced models within Pyrate to conclude what the main factors are that control diversity dynamics in different microfossil groups. To conclude, microfossil diversity definitely evolves through time. All microfossil groups were uniquely impacted by different environmental perturbations to which their response was not consistent from one extinction to the next, and most of my hypotheses that I proposed were rejected. Generally, calcareous and siliceous groups experienced opposing impacts across major transitions. Perturbations associated with warm waters and deepening of the CCD often promotes diversification in calcareous groups, while cooler sea surface temperatures, high latitudes and elevated nutrient inputs are associated with siliceous diversification. During glaciation periods, dwarfing of foraminiferal tests appear to provide a selective advantage, suggesting the possibility of sea surface temperatures being an imperative control on foraminiferal size and subsequent diversity. Despite ingrained faults involved in utilising models, Pyrate provides a leap forward in quantitative testing of diversity change throughout geological time, but also stresses the need for more robust occurrence data records, particularly within siliceous groups as well as in high latitudes. Thank you very much for listening to my talk. I'm really looking forward to answering any questions that you may have.